Bismillahirrahmanirrahim. Now we continue with the paper 4 which I did in the last two videos, and these are the three questions which were left out of that. Now starting with question number 6, figure 6.1 outlines the first three stages of respiration in aerobic conditions. Glucose to triose phosphate, and we've spent 2 ATPs. And the triose phosphate, now there are two of those, so you remember this 6 carbon glucose and 2 3 carbon triose phosphate. And then we have taken out 2 NADs, reduced NADs, and we have also earned 4 ATPs. But we've always spent 2 ATPs. So 4 minus 2 is a total net gain of 2 ATPs. And then we've made pyruvate. And then from pyruvate to acetyl-CoA, you remember this is the link reaction. Now in the link reaction, you know what is going to happen. Now they have specified in the syllabus, you must know where decarboxylation takes place and where dehydrogenation takes place. So dehydrogenation is the formation of reduced NAD, and molecule Y is the decarboxylation and the enzyme decarboxylase. So molecule Y has to be CO2. Then acetyl coenzyme A is formed, and there are two of those, 2-pyruvate, 2-triose phosphate. Just trying to make you remember it for a next question in the coming exam. Then in the Krebs cycle goes twice. The reason why it goes twice because you have 2 acetyl CoAs. Now molecule Y, and there are 4 of those. Why? Because the Krebs cycle is going to go twice, and you know molecule Y here was also carbon dioxide. And ATP2 are being made here, that is substrate level phosphorylation. Wherever ATP is made, that is substrate level phosphorylation here in the Krebs cycle and in the glycolysis. But the one taking ATP being made in the electron transport chain, or the ETC, that is called oxidative phosphorylation. And then we have 6 reduced NAD. Why? Because we have 3 NADs in one cycle. There are 2 here and there's 1 here when oxaloacetate is regenerated. So what you have to understand is, and then 2 FADs. Why? Because the FAD is going to be made somewhere here. So the FAD is going to be twice because the Krebs cycle goes twice. Now let's look at the questions. Name molecule Y. In figure 6.1 we know it's carbon dioxide. Please, when it says name, give me the whole name, not your nickname. So you can't say CO2. So carbon dioxide. Then explain in figure 6.1 shows that glycolysis involves oxidation. Now the answer to that was triose phosphate dehydrogenated or reduced NAD made. So oxidation would wherever it be reduced NAD being made. So that would be reduced NAD is the point where oxidation was taking place. At one time, it was thought, part C, that the oxidative phosphorylation of one molecule of reduced NAD results in the synthesis of 3 ATP. Yeah, we always learn that is, I always tell you to remember it like this, you know, there are 3 lines in N. So NAD gives you 3 ATP molecules. Then it could be coming in the next exam question. And one molecule of FAD gives you 2 ATPs. So this is something we know, this is some information that we always give you. Using figure 6.1, a theoretical value for the net number of ATP molecules that are synthesized for each molecule of glucose can be calculated. Modern research has shown that the actual net number of ATP molecules synthesized for each glucose respired is much lower than the theoretical value. So using figure 6.1, calculate the theoretical value for the net number of ATP that are synthesized for each molecule of glucose respired in all phosphorylation reactions. Show your working. Now that's 38 molecules, 38 ATP molecules, but how did we do the calculation? I want you to look at that. As you can see here in the mark scheme, it's very, very clear how it's been made. So ATP was 4 minus 2 because we had actually spent 2 and then we gained 4. So I gave you 2 rupees and you gave me back 4 rupees. So it's a net gain of 2. So the 2 here. And then every NAD was 4 NADs being made in glycolysis. So 4 NAD, because they've bracketed glycolysis and the link reaction together. So 4 NAD into 3 would be 12. Then in the Krebs cycle, they've said, okay, we gained 2 ATP. And then because we had 6 NAD, so 6 into 3 is 18. And then because we had 2 FAD, so 2 into 2 is 4. So that is how they've done their calculations. And that is how you were supposed to show some sort of this working to show me the value of 38 ATP molecules. You see, when you're answering such a question, which is a very theoretical question, the suggest is always something which requires a lot of, sense. Like for instance, if I asked you, what is your salary? And you said, well, my salary is 1 lakh rupees. But I said, why do you get 80,000? And you say, well, because maybe 20,000 is the tax which is deducted. So your actual salary is 1 lakh, but you get only 80,000 because you've sort of maybe the school cut some money from salary. Maybe it's a provident fund, or maybe it's some sort of thing which the government takes that money out of you. So it is saying Y is actually not 38. So we have to think of things, and then ATP is used to transport the pyruvate into the mitochondria. Now you remember glycolysis takes place in the cytoplasm, and then the pyruvate has to enter the mitochondria. So maybe that is an energy requiring process. Plus all the NADs that were made in glycolysis, they need to be also brought into the mitochondria. So that would require some sort of energy. Maybe that, maybe some protons leak from the intermembrane space. Maybe some energy is lost as heat. Maybe the glucose is not completely broken down. And or we could have said the reduced energy is used for other reactions. So, your suggest questions are technically challenging. Why? Because they check how general your knowledge of biology is and how you can cope with a new sort of a situation. And paper 4 does have a lot of suggests, so you need to be really thinking on those lines whenever you're doing a suggest question. Okay, then we come on to the part C of the question. Now this is outline the roles of NAD and FAD in aerobic respiration. Now this has come in many papers, and you just have to remember these, and you just have to sort of, I always say, just up iska ratta lagaye. Ratta means you just have to rote learn it. As I always say is, NAD and FAD, they are the chotus or the assistants or the minions for the enzyme dehydrogenase. So when we call them some sort of an assistant to something, then we call it a coenzyme. Why? Because dehydrogenase is an enzyme, which is a coenzyme, which is an enzyme. So they are the helpers of this enzyme. So we call that coenzyme. So NAD and FAD are coenzymes for helping out the enzyme dehydrogenase. Where are they helping out the dehydrogenase? In the three processes, which is the glycolysis, link reaction, and the Krebs cycle. Because that is where NADH is made. So, and what are they doing is they are helping us to transport the hydrogen, because the hydrogen is being taken out in the glycolysis, link reaction, and the Krebs cycle, and NADH. So they are the carriers, they are the transport or the carriers for hydrogen. So transports hydrogen, you can say carries hydrogen, you can say transfers hydrogen. Where? To the ETC, because that is where then it is culminated into the formation of ATP. So they go to take it to the ETC, electron transport chain, on the inner mitochondrial membrane. So, I mean, in one sentence, like you could have said, NAD and FAD are coenzymes, help the dehydrogenase enzyme to transport hydrogen to ATC. So, one sentence would get you all your 3 marks. Don't look at sentences. Please be very careful. What we're looking at is what you've written in that sentence. So, it's not the number of sentences you wrote, it is how you wrote. That is the important essence of biological English. Now, coming to the E part of the question. Rolinon is used as an insecticide, whatever the chemical they've named. So rotinon is used as an insecticide. Rotinon kills insects by inhibiting the transfer of electrons in the electron transport chain of the mitochondria. Explain how rotinon affects ATP synthesis in the mitochondria. Now the clue is that they're saying, okay, inhibiting the transfer of electrons in the electron transport chain. So if you remember ETC, now, then ETC, what is going to happen is, I'm going to revise this very quickly with you. This is the outer membrane, this is the inner membrane which is folded, and NADH comes here. And NADH gives up its hydrogen. Hydrogen splits into a proton and electron. Electrons are shuttled across the membrane, and oxygen is the final electron acceptor. But when this is happening, what has happened here also is that the hydrogen ions are being pumped. So these hydrogen ions are going to be pumped into the intermembrane space. When these hydrogen ions are going to be pumped into the intermembrane space, now they have no way to come out. Now they only have one thing, which is the ATP synthase, which is going to allow them to come out. So the ATP synthase is going to come here, and they're going to flow through. The hydrogen ions are going to flow through this, and ATP is going to be made. So it's a quick revision of ATC. So if you look at the mark scheme says no energy released. Why? Because there's going to be no ATC. So no energy or less energy released from ETC, no chemiosmosis. This whole process is called chemiosmosis. So no chemiosmosis, right? No protons pumped to intermembrane space. So this thing is not going to happen. No protons will be pumped into the intermembrane space, which I just explained to you. So no proton gradient. There's not going to be a difference. There's not going to be a lot of hydrogen here and no hydrogen here. So if there's going to be no proton gradient, then no protons will diffuse through the ATP synthase. This is not going to happen. So no ATP is going to be synthesized. So just a quick reread of the Mark scheme: no energy release, no chemiosmosis. Of course, you can say less also, less or no. So no chemiosmosis, no protons pumped to the intermembrane space, no proton gradient, no protons diffused through ATP synthase. So no ATP synthesized. To question number 7. The function of the enzyme. Now just look at the enzyme. The function of the enzyme iduronate 2 sulfatase. The name shouldn't, sort of make you upset, or just don't confuse. Just say, okay, enzyme, whatever, A, is to break down certain complex molecules. Hunter syndrome is a rare inherited condition in humans caused by iduronate 2 sulfatase not functioning properly. So this enzyme is not functioning properly. So that means it's not being made properly. If it's not being made properly, there has to be a defect in the DNA. This will cause the complex molecules to build up, leading to progressive damage to many organs. So inherited condition, defect in the gene, that gene results in a defective enzyme being made, and because of that defective enzyme, complex molecules build up and organs are damaged. Explain how a gene mutation can lead to this enzyme not functioning properly or not being produced. Now, very simple. It's a gene mutation means, what is it? It's a base deletion or a base insertion or a base substitution, or which will result in a frame shift, which will result maybe in a stop codon. So that protein is not properly made, which means that the primary structure will be affected, which will change the primary structure. And this change in the primary structure will result in a change in the 3D shape of the enzyme. So if the enzyme was supposed to be made like this, and now the enzyme is going to be made like this because there's a stop codon and all the amino acids have not been actually been put into it. Now this is a non-functional enzyme. Active site does not bind. So the active site, this is the one which was the correct one. This is the wrong one which is being made in people with Hunter syndrome. So how are the substrates going to fit? Because the active site has changed. So a quick look at the mark scheme: base deletion, frame shift, maybe a stop codon resulted. This changes the primary structure, so changes the 3D shape, and the active site does not bind to the substrate. So this is a question which you must understand. If any such question comes in the next exam, these are the mark schemes that you're supposed to be giving me. Then coming on to the B part of the question, Hunter syndrome is caused by a recessive allele located on the X chromosome. So it has to be a small a. Any letter you use, I'm not saying use the letter A. Maybe if you want to use Hunter, so you want to use the letter H, I'm fine with that. Using appropriate symbols, draw a genetic diagram to show the probability of a normal male and a heterozygous normal female having a child with Hunter syndrome. So, I mean, what information is given in the question? First, look at that. First, it's a recessive allele and it's always on the X chromosome. So, it has to be X raised to the power small h. Show the of how a normal male, so you have here a normal male and a normal female, and how could they have a child with Hunter's syndrome? See, my favorite way of making you do genetics question is that you start your question from the answer. Now they've said, how can there be a child with Hunter syndrome? So that child with Hunter syndrome has to be this, if it's got to be a female. If it's got to be a male, it's got to be this. So if you have to realize, okay, so somehow we have to get this as the answer. So if that is to get the answer, then somehow one has to be, so let's look at this now. In a normal male, he has to have X big H and Y. But in a normal female, it can be either X big H, but then you can't have this kid. So then you have to figure it out, okay, well, what can I do so that, because I have to get this allele from the mother maybe. Because if you look at, this is the answer which we were supposed to get. Now, how can you get it? But they've said they are normal people. This is in the question. They've said it's normal people. But this is the answer which you have to get. So just like in maths, if you know the answer, you can go back. I think you can do that. I'm not a great maths student, but I think that's how I used to do it when I was doing maths. So this is how you have got to come up with what is the answer we want. We want a child with Hunter syndrome. A child with Hunter syndrome has to have the both recessive alleles or one recessive allele, because they've said to you that it is a disease which is caused by a recessive allele. Understand that this question is marked per line. They've given you these headings, but you have to do it according to the headings. You might not write the headings in any other, but here they've given you the headings. So the parental phenotype is given. Now the parental genotype is this one. Why? Because they are normal. So it has to be X bada HY and X big H and X small h. Then the gametes have just got to be written separately. So you just have to write it separately and give it a little space. And then of course I like to circle them, because that is what we teach you. So just write it a little spaced out. And this is the line for the gamete. So there's one mark for this and there's one mark for this. Then the offspring genotype. So if this sperm fertilizes this ovum, we have a X big H, X big H. But then if we have this fertilizing this, then we have a X big H and X small h. Or if we have this would be X big H. We always write that first and then we write the Y. Or if it is this, then it's going to be X small h and a Y. Now, so this would be one line, one mark, second mark, and now this is going to be the third mark. So the third mark is for that. Then the fourth mark is, then you've got to tell me, okay, what are these? So this is a normal female. Then this is a carrier female. Now this is a normal male. And this, of course, is the Hunter S male. The Hunter sufferer, the Hunter person who has this Hunter syndrome, he is the sufferer. So the Hunter sufferer. But then they've also asked you, now if you look at it, they've also asked you probability. So because remember there are 5 marks for this. So how am I going to give you the 5 marks? So 1 mark for this, 1 mark, so very easy, 1 mark for this, 1 mark for this, 1 for this, and 1 for writing the correct phenotypes. And then the 1 mark is 0.25. The probability is 0.25, or if you wrote 25%, or if you wrote 1 in 4. I don't like writing 1 in 4, but that confuses you, but it's up to you. 0.25 or 25% chance of having a child, a male child with Hunter. So probability, you had to give me the probability. So this would get you your 5th mark. So I always say, genetics questions, I love genetics questions because you can get your full marks on it, because there's not much that you need to write, but you need to be very correct. Now coming to question number 8, California salamanders are terrestrial amphibians. An original population of California salamanders occupies an area of forest in northern California. Approximately 10 million years ago, 2 separate populations, A and B, started to migrate from the original population. Now you can guess which chapter there's now all about. Population A traveled along the coast of California to the west of the Great Central Valley, and population B traveled east of the Great Central Valley. The 2 populations now live close to each other in Southern California. And figure 8.1 outlines the migratory routes of populations A and B. So original population they're showing you here, and then they've shown you the migration route. So one migration route is this one, which is on the right side of the valley, and then one immigration is on the left side of the valley. So there is the Great Central Valley and then the migration route, and then they have come down here. Now population A now is here, this part, and population B is now here in this part. So migratory, now they live close together. So now they live close together, but originally they were original population, and now so you see how the question is being worded now. Now coming to the part of the question. Salamanders from population A rarely interbreed with salamanders of population B. If they do interbreed, the offsprings are infertile. Again, thinking now back, we know the definitions becoming, they're asking us on that. Suggest and explain the sequence of events that have resulted in these two populations becoming reproductively isolated from each other. So very, very easy question, and there's some mark scheme points which you can write for every such question. Now let's look at a few salamanders. Now you can see these different types. They've got these different colorings on it. There's a tiger, there's a marble, there's a fire, there's blue spotted. Then, just giving a look at it gives you the feeling of the mark scheme. What is possibly going to be the mark scheme? The mark scheme is going to be very simple. Now, due to the Great Valley, during the migrations, they were separated, geographically isolated. So maybe these ones were separated from these ones. So maybe these ones were separated from these ones. So no mating, no breeding. No mating, no breeding. So different selection pressures. Maybe these were smaller and they ate less, so they survived. So different selection pressures. Why? Because now they were different habitats. Maybe the food available to these was more, to these was less, so these ones probably finished off. So different mutations occurred. Natural selection took place. Genetic drift. Genetic drift means a few separate and then they start the population. Founder effect. Genetic drift or founder effect, both is one mark scheme point. Now this would result in change in allele frequency. Allele frequency means maybe these were found as well, but now only these were found as well. Now these are only found, so the color is different now. So change in allele frequency then leads to different morphological or behavioral or biochemical features. Why? Because now these were maybe smaller in size, so these survived. So leads to different features being suited to the environment surviving. And then of course, what is this called? This is called allopatric speciation. Allopatric means that they are separated. Alag ho gaya, I call that in Urdu, but of course that's my native language. But allopatric means the name given to this separation when they are geographically isolated. So please understand this question in the context of any animal. This picture gives you a very good idea of how maybe these were better advantage, maybe these were at a better advantage, these were not at an advantage. So just once again the recap of the mark scheme. Due to Central Valley, during migration, populations are separated, geographically isolated, no mating, gene flow between these populations, different selection pressures or environment or habitats, different mutations. This is natural selection. Genetic drift or founder effect would result. This would cause change in allele frequency. These are very technical words. If you just remember these words, you can write it for any such question in the exam. That's a very tricked way that you can learn how to do. Change in allele frequency, or you said change in the gene pool or the entire gene pool becomes different, leads to different morphological, behavioral, biochemical, physiological features being shown in the new populations. And of course, the technical name for this is allopatric speciation. Allopatric speciation. Now, there's something that you must know. This is a technically biological word. That finishes this paper, because you see the essay questions now have been removed from the syllabus. You're not going to get any essay questions. So that one little choice that you had is now over. So please do not start, revising the essay questions. You can go through them at times just for the for increasing your knowledge, but otherwise I would not like you to do them. So this finishes this paper, and hopefully I should be continuing with a few more paper 4s and uploading them on a frequent basis. And thank you once again.